The equipment required for growth curve preparation includes Bunsen burner to sterilize the things, hemocytometer, इसके बारे में हम बात करेंगे, to count the cells, incubator to incubate the inoculated cultures, microscope to observe the cells. Then in order to shake the culture flasks, we need rotatory shaker at 30 degrees centigrade, spectrophotometer and vertex mixer. This figure shows you this vertex mixer. The vertex mixer is basically meant to shake or mix the cell population which could have settled down at the bottom of the test tubes. Rotary shaker is meant to shake the culture flasks at a given speed so as to equally distribute oxygen in the culture media. As we are going to compare minimal medium and the complex medium and their growth potential for the specific microorganism, we have to set up the cultures. For that purpose, we can set up a microorganism culture in relatively smaller volume. For example, 50 milliliter of the culture medium can be inoculated with the given microorganism, and then following the growth conditions, incubate it at a given temperature for a given time period with the shaking conditions, vigorous shaking conditions. Next day, using the overnight cultures, we can make a relatively larger volume. We can inoculate a relatively larger volume of the culture medium so as the density of the cells or the number of the cells should be decreased relatively. As for example, in this diluted culture, we should have the cell population somewhere around 5,000 cells per milliliter in comparison from the stock which had the 50 million cells per milliliter number. Again, these diluted samples are going to be incubated at the growth conditions, in this case 30 degrees centigrade with the vigorous shaking, and after that we have to remove 5 milliliter of the culture medium at regular intervals. We can decide the time period at our own choice, but in this case we are going to remove the culture medium from the growing culture after 4 hours, 8 hours, 12 hours, 16 hours, up to 36 hours. And these culture media, they are going to be stored in the refrigerator until further use. We will use these culture media which were taken at regular intervals from the growing culture to set up a growth curve.